Hello, everyone. I welcome you once again to another edition of Hematology and Blood Transfusion Science Lecture Series by Dr. Ifeyinwemeri and Okafor, a lecturer in the Department of Hematology and Blood Transfusion Science, College of Medical Sciences, University of Calabar, Nigeria. For some time now, we have started lecture series on iron metabolism. We have done the first two lectures on iron metabolism, iron absorption, and today we are going to be looking at regulation of iron homeostasis. Regulation of iron homeostasis. If you have missed our previous lectures on iron metabolism, please feel free to go to our channel and listen to those lectures because it will help you to understand what we are doing today better. And if you have not subscribed to our channel, feel free to subscribe to our channel. Thank you very much for your support. Like I said, today we are going to be discussing regulation of iron homeostasis. Regulation of iron homeostasis, and it is expected that at the end of this lecture, you should be able to explain the mechanisms that are involved in maintenance of iron homeostasis in our system. You should also be able to know the primary regulator of iron homeostasis, and should be able to explain the role of hepcidin in regulating iron balance. The role of hepcidin in regulating iron balance. In our previous lecture, we discussed iron absorption, and we said that the iron that we eat in our food is in form of is in two forms. The heme iron is in form of hemoglobin, myoglobin, that we get primarily from animal food, consuming meat, fish, poultry source like chicken, egg yolk, and we have non-heme iron. Non-heme iron is iron that we get from our meals when we consume food like cereals, vegetables, legumes, grains, that are rich in iron. So basically, we have the two forms of iron in our diet, the heme iron and non-heme iron. And these two forms of iron are absorbed differently. But the point of absorption is in proximal duodenum. In the proximal duodenum, the iron is absorbed in the proximal duodenum. The heme iron is transported across the luminal wall of duodenal enterocyte by what we call heme transporter. Heme transporter. Heme carrier protein 1, heme transporter. It is transported as heme. But our non-heme iron is absorbed across the luminal wall by what we call divalent metal transporter 1. Divalent metal transporter 1. The food iron that we eat is in form of heme iron and non-heme iron. Our non-heme iron, our heme iron is absorbed directly through heme transporter, but non-heme iron you have here in our meal occurs in form of iron 3. Remember I said earlier that iron has these peculiar properties of being present either as iron 3, ferric iron, and iron 2, ferrous iron. And it has the ability to change from ferric to ferrous and ferrous to ferric in the system. So the one we have in our food as non-heme iron is in form of iron 3. And for it to be absorbed in the proximal duodenum, it must be reduced to iron 2. And we have enzymes, ferroreductases on the luminal wall. Example of one of these enzymes is cytochrome B. Cytochrome B help in reducing iron 3 found in our diet in form of non-heme iron, so iron 2. And when this is done, this iron 2 can now be absorbed through a transporter, a carrier transporter called divalent metal transporter 1 that helps to absorb iron 2 into the duodenal cells. And then when this is done, the iron 2 enters into the same pool with heme iron that have been previously absorbed through heme transporter. Now, when they are inside the duodenal cell, two things happen. Is either they are stored in mucosal ferritin as part of the mucosal ferritin in duodenal cells, or they move through the basolateral walls of the duodenal enterocyte through a channel called ferroportin. The ferroportin is the channel that allows iron allows iron efflux through many cells that contain iron. Now, this ferroportin allows this iron to move into the circulation to be bound to transferrin. Remember, transferrin is the protein that transports iron in plasma. But before iron 2 can be carried by transferrin to be taken to where it is needed in our body, this iron 2 must be reoxidized back to iron 3. And we have enzymes on the walls of the enterocyte, the duodenal enterocyte, that helps in reoxidizing iron 2 to iron 3. And the enzyme is called hepstin. When this enzyme is in plasma, it is called steroloplasmin. When found in placenta, it's called cyclopen. So here we have it as hepstin, converting the iron 2 back to iron 3 before it is bound to transferrin. Once bound to transferrin iron 3, it will now pass through normal blood circulation, blood vessels, and get to where it can be utilized, is either in the erythroid marrow or in the liver where it is stored, or in the liver where it is stored as ferritin, okay? That is an overview of iron absorption. The iron is absorbed at proximal duodenum, and we have enzymes that first of all reduce our iron 3 to iron 2 before it is carried by divalent metal transporter across the luminal wall and then pass through the ferroportin channel and reoxidized back to iron 3 by hepstin before it is bound to transferrin that can carry it through the blood vessels to where it is utilized, either in the bone marrow for erythropoiesis or stored in the liver or for other processes that iron is involving in our system. Now, mucosal intelligence shows that there is a close relationship between iron need on one hand and iron dose. And also the amount of iron that is absorbed. There is a close relationship between iron need and iron dose and the amount of iron that is there absorbed. So when our system is in need of iron, like I said earlier, that iron deficiency or active erythrophoresis upregulates iron absorption, while iron overload downregulates iron absorption. And we also say that substances that stabilizes iron 3 in our diet reduces iron absorption, while substances that stabilizes iron 2 increases iron absorption in our diet. Because, you know, for iron to be absorbed, non-heme iron to be absorbed, they must first of all be reduced to iron 2 before they can be absorbed. Okay? Then the body has 3 unique mechanisms for maintaining iron balance and preventing iron deficiency and iron overload. The body has, you know, basically 3 mechanisms for maintaining iron balance and preventing iron deficiency on one hand, and then iron overload on the other hand. First, there is continuous reutilization of iron from catabolized erythrocytes in the body. Like I said, when we are treating iron absorption, that, you know, for iron balance, for iron homeostasis, The iron homeostasis is predominantly resting on, these four variables: the nutritional intake, that is the nutritional intake / the absorption, the iron loss on one hand, the current demand of the patient or an individual, and then the amount of iron that is recycled within the body. So the body has these mechanisms to maintain iron balance. First, there's a continuous utilization of iron from catabolized erythrocyte in our body. Like I tried to introduce in our previous classes, that red blood cells, when they circulate within our body for a period of 120 days, that is the lifespan of red cells, they are pulled out of the system by the spleen, reticuloendothelial system by the spleen, and then these aged red cells are destroyed. And in catabolism of red blood cells, hemoglobin is destroyed, and hemoglobin is made up of heme and globin. The heme aspect of it is now catabolized, and you have protoporphyrin ring, and then iron. The protoporphyrin ring is what is degraded to bilirubin, giving you the bilirubin we have when we have hemoglobin catabolism. And then the iron part of it is recycled and reused. So there is continuous internal supply of iron from catabolized erythrocyte, from aged erythrocyte that has been destroyed, and the iron in it reused. Again, so the body tries to maintain iron balance by reusing, the iron that is gotten from catabolized erythrocyte that has been aged and then destroyed, and the iron recirculated or reused. Then the second system is the access of specific storage protein like ferritin, which can store and release iron to meet excessive iron need. Our system has the ability to have iron stored. Remember, we said that there are two proteins that are involved in iron storage. We have ferritin and then hemosiderin. So when we consume excess iron, when our body, we have the capacity to take in more iron in our diet, or that our system stores this iron as ferritin and hemosiderin, so that if for any reason we are not able to take in as much, the body can now use those that are in the storage facility and prevent us from having iron deficiency, prevent the system from having iron deficiency. So to maintain iron balance and to prevent the system from having iron deficiency, one, there is a continuous utilization of iron from catabolized erythrocyte in the system. Two, there is stored iron as form of ferritin that the system can always fall back on when in need of iron. Third, there is also regulation of absorption of iron from the intestine, with an increased iron being absorbed in the presence of decreasing body iron stores, and a decreased iron being absorbed when iron stores increases. There is also a tight regulation of iron absorption from the intestine. Iron is absorbed more when the body is in need of iron, and then there is decreased absorption when the iron stores is increased. When the iron stores are depleted, the body absorbs more iron from the diet. When iron stores increases, the body reduces its absorption from the diet. Now, at this point, regulating iron absorption at the point of intestine, that is where iron is being absorbed at proximal duodenum, and also releasing of iron that has been stored in the ferritin, storage proteins like ferritin and hemosiderin, is being tightly regulated by a protein called hepcidin. Is being tightly regulated by a protein called hepcidin. That is the ability of the system to regulate iron absorption from the intestine, and also the ability of our body to release iron from stored proteins like ferritin and hemosiderin, is being tightly regulated by a protein called what? Hepcidin. Now, hepcidin is the primary regulator of iron homeostasis. The protein called hepcidin has been found to be the primary regulator of iron homeostasis and is presently regarded as the key to the iron balance regulator. The protein hepcidin is primarily regarded as the key to the iron balance regulator. Hepcidin is made up of about 20 to 25 amino acids in length and has 4 disulfide bonds. It's made up of about 20 to 25 amino acids in length and has 4 disulfide bonds. It is encoded by a gene called hepcidin antimicrobial peptide gene. This gene is located on chromosome long arm of chromosome 19, position 13. This gene is located on the long arm of chromosome 19. Hepcidin is the primary regulator of iron homeostasis and is presently regarded as the key to the iron balance regulator. Remember what I said here, that the body has 3 unique mechanisms for maintaining iron balance and preventing iron deficiency and iron anemia. 1, the body has the capacity to reutilize iron from catabolized erythrocyte. The body has the ability to get iron from stored iron, to fall back on iron that is stored in the system as ferritin and homocysteine. 3, the body has the capacity to regulate iron absorption from the intestine, that is the proximal duodenum, when the body is in need of iron more, and also reduce the absorption when the body does not need iron. But to do that, to absorb more from proximal intestine, proximal duodenum, and also to release iron from the stored iron like ferritin and homocysteine, depends on a protein called hepcidin. In the diagram that we presented earlier, we said that during absorption, iron in form of iron 2 is released into the circulation by passing through a channel called ferroportin. This ferroportin allows iron that has been absorbed through the proximal duodenum to move into the blood vessels so that they can be bound to transferrin. But remember, the iron in form of iron 2 must first of all be converted back to iron 3 in the presence of hepcidin before it can be bound to plasma transferrin. Now, if you look at this channel here, the iron bound to plasma transferrin can be taken to erythroid cells for production of erythrocytes, and they can be stored in the liver. But there's another thing here. The liver also produces another protein called hepcidin. This hepcidin has the capacity to block this channel called the ferroportin. So hepcidin is at the center of regulation of iron homeostasis. When the system is in need of iron, the liver, the production of hepcidin is reduced. So this channel is left open for the body to absorb more iron through the proximal duodenum. But when the body has enough iron stored, the liver produces more of hepcidin, and hepcidin blocks this channel ferroportin, and what? The iron absorption for the proximal duodenum is reduced. Okay, and I said that this hepcidin is encoded by the gene hepcidin antimicrobial peptide gene, which is located on chromosome 19. Now, hepcidin acts as a negative regulator of iron stores. Hepcidin acts as a negative regulator of iron stores. In response to increased iron levels, when the body, need more iron, when there is increased iron levels, when there is increased iron level in the system, this increased iron level can be due to increased intake through diet, supplementation, or blood transfusion. when our body is in a state of high iron balance, there's an increased amount of iron in the system, increased intake of iron in the system, which might be due to, increased intake through our diet, through supplementation, through blood transfusion. Then the liver increases hepcidin synthesis. The liver increases hepcidin synthesis. And when this is done, this hepcidin will now act on 3 sites at absorption sites. This absorption site is at the duodenal enterocyte, at the point of proximal duodenum, where iron is absorbed from the diet. Remember the diagram that we showed earlier? The ferroportin there, the hepcidin blocks that channel, and iron is no longer absorbed through the duodenum. The hepcidin can also act on storage sites like ferritin found in hepatocytes, the liver cells, and block the channel through which ferritin releases iron to the circulation, which is also ferroportin. It can also act on the recycled iron. Macrophages, which helps in destruction of aged red blood cells, contains ferritin and hemosiderin, where the recycled ions are stored. And they have this channel ferroportin too. So the hepcidin can also block the channels and macrophages, the ferritin and hemosiderin found in macrophages, and stop them from releasing recycled iron into the system. That is when in response to increased iron level in the system. When there's increased iron level in the system, the liver increases hepcidin synthesis, which act on absorption sites, blocking the ferroportin channel there. Can also act on storage sites like ferritin found in hepatocyte, stopping them from releasing more iron into the system. It can also act on macrophages in the spleen, and stopping them from releasing recycled iron from the system. Remember, the body gets iron from 3 different sources, and like we discussed earlier, from absorption from our meals, from recycled iron gotten from destroying this catabolized red cells, and also from iron that has been stored as ferritin. So when there is increased iron in the body, hepcidin is produced by the liver to block these channels from bringing in more iron. It can act on duodenum, stopping the duodenum from bringing in more iron from the diet. It can also act on the liver cells, stopping the ferritin that is stored in the liver cells from releasing more iron into the system. It can also act on the macrophages, stopping the ferritin that is stored in the spleen from releasing more iron, recycled iron, into the system. Hepcidin exerts its influences by binding to and inducing the internalization and degradation of ferroportin, the only known exporter of iron. Ferroportin is the only known exporter, only known channel through which iron is exported into the system, either at proximal duodenum or at ferritin, or at ferritin in the hepatocyte, or ferritin in the macrophages and the spleen, reticuloendothelial system. So hepcidin regulates iron by doing what? Exerting influence by binding to and inducing the internalization of and degrading of ferroportin, which is the only known channel for iron exports. Ferroportin mediates influx of iron from iron stores like ferritin in the hepatocyte, ferritin and hemosiderin in the macrophages, and then from iron that is absorbed from the diet. Ferroportin is negatively regulated by hepcidin. Looking at this diagram here, regulation of iron absorption, intracellular and extracellular iron concentration when it increases. When it increases, excuse me, when intracellular and extracellular iron absorption increases, or when the inflammatory processes in our system increases, these are triggers for production of hepcidin. So the liver cells produces more of hepcidin, and this hepcidin goes to the duodenal enterocyte and blocks ferroportin channel, which is the channel that allows iron absorption from the diet into the circulation. It also goes to the splenic macrophages and block ferritin and hemosiderin that are stored in splenic macrophages, blocking the channel through which this ferritin releases iron into the system. It also stops the hepatocytes, the ferritin that are stored in the hepatocytes, from releasing iron into the plasma. Remember, during iron absorption through the proximal duodenum from our diet, iron moves from our diet in form of non-heme iron and heme iron reduced to iron 2, and then through the ferroportin into the plasma ion, where it is incorporated into transferrin. They must pass through the channel called the ferroportin. An increase intracellular and extracellular ion upregulates hepcidin production, inflammatory processes, or production of interleukin 6 and interleukin 1 upregulates hepcidin production. And once hepcidin production increases, it acts on ferroportin, stopping duodenal, proximal duodenum from absorbing more iron from our diet. It also acts on the ferritin that are stored in the splenic macrophages. And this has to do with recycled iron, because most of the recycled iron that is gotten from catabolized erythrocytes, are stored in the ferritin in the spleen of macrophages, ferritin and hemosiderin in the macrophages. And they also contain ferroportin. And ferroportin is the channel that allows this ferroportin to release iron to the circulation. So increased extracellular iron also increases hepcidin production. And this hepcidin production acts on this site on splenic macrophages, stopping them from releasing the iron that is stored in them. It can also act on ferritin found in hepatocytes and also stopping them from releasing iron found in them into the circulation. But erythropoietic iron requirement, when there is iron needs in the system, maybe because of anemia, iron deficiency anemia, or increased erythropoiesis is needed in the system, then this causes downregulation of hepcidin production. The hepcidin production is downregulated, and the ferroportin is open. The channel is opened up for what? Iron absorption through the duodenal, proximal duodenum, and then the splenic macrophages are allowed to release iron through the ferroportin channels. The same thing with hepatocyte. The ferritin stored in hepatocyte are also allowed to release iron into the plasma ion through the ferroportin channel. Hepcidin is production of hepcidin is downregulated in erythropoietic iron, increased erythropoietic iron requirement. And then these three sites are allowed to release iron to the plasma. Ferroportin is a transmembrane protein composed of monomer dimers present in hepatocyte. You can find it in hepatocyte and then enterocytes, duodenal enterocytes, macrophages, spleen, and bone marrow, which regulates the amount of iron present in the extracellular and intracellular medium. Ferroportin serves as the receptor for hepcidin. When hepcidin binds to ferroportin, the latter transport molecule is internalized and undergoes proteolysis. As a result, the capacity of cells to export iron is decreased. So hepcidin exerts its influence as iron regulator of iron homeostasis by acting on ferroportin, which is only known channel through which iron is exported out to the plasma, either absorbed iron, recycled iron, or stored iron. By acting on ferroportin and stopping it from allowing iron to be released into the system. There are factors that modulate the transcription pathway of hepcidin antimicrobial peptide gene. Remember I said earlier that hepcidin antimicrobial peptide gene is the gene that found on chromosome 19, is the gene that encodes for the production of hepcidin. And there are certain things that actually modulate the transcription factor that modulate the transcription pathway of this HAM gene, HAMP gene. They are like hypoxia, iron concentration, erythropoietin level, inflammation, anemia, and among others. These factors activate, they do this, they modulate the transcription pathway, this gene. They do this by activating two major pathways. These factors activate two major pathways that act directly on HAMP gene. And this pathway is the first one is signaling pathway occurs through bone morphogenetic proteins, and SMAD pathway. The signaling pathway that involve bone morphogenetic proteins and SMAD signaling pathway. And the second pathway is the genius kinase signal transducer and activator of transcription signaling pathway, JAK/STAT pathway. The first pathway that is involved in modulating the HAMP gene transcription pathway is the BMP-SMAD pathway and JAK/STAT pathway. The JAK, we have the full meaning, the genius kinase signal transducer and activated transcription pathway, while the BMP is bone morphogenetic proteins and suppressor of mothers against decapentaplegic pathway. The BMP/SMAD signaling pathway is a major transcriptional regulator of hepcidin expression that controls hepcidin production in response to most of its known regulatory signals. Most of these known regulatory signals include hypoxia, erythropoietin, inflammatory processes, even increased upregulation of interleukin-1 and interleukin-6. The studies have shown that BMP/SMAD signaling pathway and JAK/STAT signaling pathway interact to induce hepcidin during inflammation. They interact to induce hepcidin during inflammation. So through inflammatory processes, production of hepcidin is increased. And when this is done, the release of iron from the stored iron or absorption of iron from diet or from the recycled iron is reduced. And during those inflammatory processes, maybe some school of thought has said this is also part of the body's mechanism to withhold iron from invading organisms that cause the inflammatory processes in the first instance. Studies have also shown that mutations and polymorphisms that has to do with hepcidin antimicrobial peptide gene, human homeostatic iron regulation gene, hemojuvelin gene, and phenylalanine gene, and also transmembrane protein serensis gene. The mutations that occur in these genes influence serum hepcidin concentration. Mutations that occur in this gene influence serum hepcidin concentration. The protein human homeostatic iron regulation gene, HFE protein, acts on the regulation of hepcidin transcription through its interaction with the transferrin receptor. And research has shown that patients with juvenile hemochromatosis who have mutations on hemojuvelin manifest low hepcidin level. Now, in our previous lecture, we talked about the relationship between transferrin and transferrin receptor. And we said that transferrin, the only work of transferrin is to transport iron in plasma and take them to the point where iron is being utilized. But transferrin cannot take iron within the cell. Therefore, transferrin needs to bind with transferrin receptors that are found on the surface of the cells that need iron. and help to internalize this ion within the cell so that ion can be utilized. Therefore, because of the role of transferrin receptor in the binding and endocytosis of dipheric transferrin, control of transferrin receptor biosynthesis is also a major mechanism for regulation of ion metabolism. The studies have also shown because of the involvement of transferrin receptor in internalizing in the endocytosis of dipheric transferrin, controlling biosynthesis of transferrin receptor has also been shown to also be a mechanism also for regulation of ion metabolism. Intracellular regulation of ion metabolism. Now, within the cell proper, the synthesis of ion regulatory proteins like apoferritin is ferritin without ion, transferrin receptor, diamino ribulinic acid synthase, diamino ribulinic acid synthase, apoferritin, aconitase, divalent metal transporter 1, and ferroportin is regulated post-transcriptionally. The messenger RNA for each of these proteins contain one or several ion-responsive elements. The intracellular ion pool is regulated by binding of ion regulatory proteins 1 and ion regulatory proteins 2 to ion responsive elements. The intracellular ion pool is regulated. By binding of ion regulatory protein 1 and ion regulatory protein 2 to ion responsive elements. Remember what I said earlier, that the synthesis of apoferritin, transferrin receptor, diamino ribulinic acid synthase, apotransferrin, aconitase, divalent metal transporter 1, and ferroportin, they are all regulated post-transcriptionally. And then that the messenger RNA for each of these proteins contains one or more several ion-responsive elements. That the intracellular ion pool, that is intracellular ion pool, is regulated by binding of ion regulatory proteins and ion regulatory proteins 1 and 2 to ion responsive elements. Now, ion regulatory protein 1 and ion regulatory protein 2 are RNA binding proteins that interact with ion responsive elements to control the translation of proteins involved in ion metabolism. Control response of proteins that are involved in ion metabolism. The ion responsive elements exert their effect by binding one of the 2 ion regulatory proteins. The ion regulatory protein 1 is cytoplasmic aconitase with 4 ion sulfur clusters and the ability to bind ion, which is required for its aconitase activities. The ion regulatory protein 2 is highly homologous to ion regulatory protein 1, but differs by the presence of 73 amino acid insertion in the N-terminus and the lack of aconitase activity. In absence of ion regulatory protein 1 binds to ion responsive elements, but in its presence becomes a cytoplasmic aconitase. Ion regulatory protein 2, on the other hand, is proteolyzed in the presence of ion. Studies have shown that nitric oxide has an effect on ion responsive elements, ion regulatory protein system. Remember, the ion responsive element and ion regulatory protein system helps in intracellular regulation of ion metabolism. Ion responsive element and ion regulatory protein 1 and 2 helps in intracellular regulation of ion metabolism. It increases binding of iron-responsive element. That the nitric oxide increases binding of iron regulatory protein 1 to iron-responsive elements and enhances the regulation of iron regulatory protein 2, therefore exerting contradictory effects on the regulation of protein synthesis. The effect of binding of iron regulatory proteins to 3' of iron-responsive element is to increase the stability of messenger RNA and thus to enhance the synthesis of the gene product. Conversely, when the iron content of cytosol is high, the displacement of iron regulatory proteins from the iron-responsive element leads to decreased synthesis of these proteins. Iron, the body does not really have the physiologic means of excreting iron from the system. Iron is so important in our system that the body tries as much as possible to conserve iron because the system cannot afford not to have iron at any point in time. Therefore, the body conserves iron with remarkable efficiency. That's why we have the iron stores in form of ferritin and hemosiderin, so that at any point in time, even when you're not taking enough in your diet, you can fall back on what you are stored in your system because the system cannot afford not to have hemoglobin. Remember, we said that blood is life, and hemoglobin is that protein that transports oxygen throughout the system, among other functions that it does. So the body cannot afford to not to have hemoglobin. So at any point in time, the body needs iron to continue the synthesis of hemoglobin. So the body conserves iron as much as possible by having iron and stored iron, and also having to recycle senescent erythrocyte, catabolize erythrocyte. The iron is there, recycled. Okay. Most iron loss occurs by way of the body. The system loses iron through this schemated intestinal cells in the feces. Less than in the feces, the body, through the feces, the body can lose about 1 milligram iron daily. And then exfoliation of skin and dermal appendages, dead skin cells, and then through urine and sweat, the body can also lose small amounts of iron, like 1 milligram. And then women who are undergoing doing lactating processes, like during lactation, the body can lose iron through milk, and then through menstruation. Women that are menstruating tend to lose more iron than those that are not menstruating. So you find that most times men don't really have a physiologic means of losing iron except through sweat or if they have acute blood loss. So most times it's always good to advise that once in a while we can go for blood donation to also help our system to rejuvenate. Also go for iron blood donation to help to our system to rejuvenate. Okay. I know that you've been able to learn 1 or 2 things from today's lecture. We said that body does everything possible to maintain iron balance because our system cannot afford not to have enough iron. But in spite of that, iron absorption must be regulated. And the main protein that is involved in iron regulation is a protein called hepcidin. And hepcidin does this by exerting its pressure on an ion channel called ferroportin that allows iron exflux either from during when iron is being absorbed from the proximal duodenum or when iron is being released from ferritins in the hepatocyte or from ferritin and hemosiderin in the splenic macrophages. It helps to block those channels, hepcidin, when the body has increased iron level in the system. But when the iron level in the system is low, the production of hepcidin is down-regulated, and then this channel's ferroportin is opened up, and more iron is absorbed through our diet, or also released from the stored iron ferritin in the hepatocyte, and also ferritin and hemosiderin in the splenic macrophages. And also said that this hepcidin is encoded by the gene hepcidin antimicrobial peptide in chromosome 19, long arm of chromosome 19. And there are different pathways that actually help in signaling the modulation of this gene, like BMD, SMAD, a signaling pathway, and JAK, STAT signaling pathway. And some of these signaling pathways are modulated by certain factors like hypoxia, erythropoiesis, inflammatory processes in our system, especially increased level of interleukin-1 and interleukin-6 helps in also causing the gene to increase the production of hepcidin in the system, and in other words, reducing iron absorption during inflammatory processes. In addition, there are also other proteins like transferrin receptor that is involved in internalizing iron into the cells. Because of their role in helping in endocytosis of dipheric transferrin receptor complex into the system for iron to be utilized within the cells. They're also involved in regulating iron balance. We also say that mutation that happens in hepcidin antimicrobial gene and other genes that are involved can also help in regulating hepcidin production, and also help in regulating hepcidin production. The system does not really have any physiologic means of losing iron, but we lose iron through sweat, a little bit of it through feces and urine, and exfoliation of dead skin. Or for women that are menstruating or lactating mothers, they can also have increased loss of iron through those means. But the body at any point in time has a tight regulation and a tight way of conserving iron in the system, and the system cannot afford to lose much iron. For more readings, you can also look at our references and also can read more on that. Thank you very much. I believe that you have learned one or two things from today's lectures. Feel free to send in your feedback. Your questions will be most welcome. Thank you, and thank you.